Hello, this is Jack from PhysiologyZone, and in this third part on skeletal muscle, we're going to be looking at muscle contraction. So we'll cover two things in this tutorial: the contraction cycle, and therefore the contraction part of the excitation-contraction coupling that you'll read about in textbooks, and then we're going to finish by looking at the length-tension relationship, which is intimately linked with the contraction. Interestingly, prior to the mid-1950s, it was a widely held belief that muscles contracted by folding themselves up, and so they lengthened and shortened by effectively folding and straightening process. And this was so hard to dispel because we didn't really have powerful enough microscopes to actually see what was actually happening. But in 1954, there were two separate teams of scientists that unbelievably published a paper on the same day in the same journal, Nature, which proposed a new method. On one team we had Andrew Huxley and Rolf Niedergerk, and they used a type of interference microscope which utilises two separate beams of light. And in the second research group we had Gene Hansen and Hugh Huxley, who used the electron microscope to study muscle. Now, fundamentally what both these teams showed was that in a contracted and relaxed state, the lengths of the thick and thin filaments within a myofibril remained the same. So effectively it was dispelling that the thick and thin filaments were folding in any way, and what was actually happening was that they were sliding over one another, and this was the responsible factor in causing contraction, thus shortening of our muscles. And this fundamental shift in our understanding of how muscles contract and relax is known as the sliding filament theory. So that leads us on to the contraction cycle, which describes the steps by which the striated muscles, so meaning skeletal and cardiac muscles, contract. So on screen you can see a diagram of myosin and actin on the respective thin and thick filaments that we've gone over previously. So firstly, for clarity, let's just go through the structures that are on screen. So here we have the thin filament with the actin molecules on them, and they have the myosin binding sites. We have troponin, and remember that troponin C is the bit that we're really concerned with for contraction, and we have the tropomyosin. Remember, the troponin and tropomyosin form a complex. So we also have the thick filament, and on here we have the myosin head and tails, and we also have proteins at its base that we've mentioned previously, but they're not particularly important to detail here. And at the top we also have calcium, which you'll remember from the last tutorial is released from the sarcoplasmic reticulum and moves into the sarcoplasm. And it's the movement of the calcium into the sarcoplasm, which is effectively represented on here as the white background, that leads to the contraction cycle beginning. And this can be broken down into 5 steps. So firstly, calcium binds to troponin C and causes a conformational change. That causes troponin to slide the tropomyosin off of the binding sites on the actin molecules. Secondly, we have cross-bridge formation. So myosin has ADP, which is adenosine diphosphate, and PI, which is just meaning an inorganic phosphate, that's on it from a previous contraction cycle. So the presence of this phosphate group phosphorylates the myosin head, and that causes a conformational change in its shape. So it attaches to the actin at its myosin binding sites, and thus it forms our cross bridges. Next we have the power stroke. So this is the bit that causes the movement. And here we get the release of the ADP and inorganic phosphate from the myosin head, and that causes another conformational change, so that the myosin head actually bends to 45 degrees, and that pulls the actin molecule towards the center of the sarcomere, which you remember is called the M line. The fourth point is that ATP comes and binds on the myosin head's actin binding site, and it releases the myosin from the actin. And then finally we have ATP hydrolysis. So as well as an ATP binding site, the myosin head also has an ATPase enzyme, and this hydrolyzes the ATP to ADP and an inorganic phosphate, and that releases the myosin head down, and it puts it into what's sort of a cocked state. So it puts it into a position where the myosin head is then ready again to bind to the myosin binding sites. And this process just repeats itself so long as you have enough ATP available and the calcium concentration near the thin filament remains high. Now, if you pull this out, it's easier to see that what's happening here is that the myosin is effectively walking its way along the actin molecule. Now, this occurs really, really fast. So there's 500 cross bridges per thick filament which attach and detach 5 times a second. So at any one time, you've got some myosin heads that attach to actin and some generating force. Others are detached and others are just ready to begin the process again. So this causes this walking along the actin molecule, pulling all of that thin filament towards the center of the sarcomere. If you extrapolate that out to the muscle fiber, you've got lots of sarcomeres lying parallel to one another in a muscle fiber. So if they all contract, the muscle fiber contracts. And obviously we have many muscle fibers making up an individual muscle, so hence that's why your muscle contracts. So the final bit is that once the stimulus for the contraction has ceased, then the calcium moves out of the sarcoplasm, and it does this as follows. So there are active transport pumps on the sarcoplasmic reticulum, and these pumps use ATP, which binds to the pump and is hydrolyzed to ADP and an inorganic phosphate. And that process causes a conformational change in the active transport pump's shape, so meaning that they open. Once they are open, they literally pump calcium back out of the sarcoplasm into the sarcoplasmic reticulum, and that returns the low level of calcium in the sarcoplasm. And as a quick side note, within the sarcoplasmic reticulum, there's a protein that's called calsequestrin, and that binds up about 20 calcium molecules each. And that's how the sarcoplasmic reticulum keeps so much calcium ions within it. So if we take this back down to the level of the thick and thin filaments, so we can see that the reduction in calcium when it's moved back into the sarcoplasmic reticulum, that causes the troponin-tropomyosin complex to slide back into the position that it was resting in originally, blocking the myosin binding sites on actin and thus preventing further contraction. So if you pull that out to the sarcomere, then we get the relaxation and lengthening of the actual sarcomere as an entity. Therefore, the muscle fiber is going to lengthen, and ultimately our muscle is going to lengthen and therefore relax. But when a muscle contracts, it doesn't always lengthen and shorten, and that's why I want to talk about now the length-tension relationship. The length-tension relationship states that the forcefulness of the muscle contraction depends on the length of the sarcomeres within a muscle before contraction occurs. So, just for interest, the optimal length of a sarcomere for maximum muscle contraction power is about 2 to 2.4 micrometers. This is because the overlap between the actin and myosin molecules is optimal at this point, so there's enough myosin overlying the actin to attach and pull it towards the center. If a sarcomere is too stretched, then there is little overlap between the myosin and actin, which therefore means that fewer cross bridges can be formed and that less tension is developed. And equally, if a sarcomere is too short, then there is plenty of overlap between myosin and actin, but the Z discs at the end of the thin filaments crumple the myosin heads, so they can't attach to the actin. And for any of the myosin heads that can attach to the actin, there's just nowhere to pull the actin because it's blocked by the Z discs. So you can effectively show this process on yourself. If you imagine flexing your wrist, that will decrease the length of the sarcomeres within the muscle. So if you try and make a fist in the flexed wrist position, you'll find it's very weak. If you then pull your wrist into a neutral position and make a fist as strong as you can, you'll find that you can produce quite a lot of force. And if you then extend your wrist all the way as much as you can, that is therefore lengthening the muscle and therefore lengthening the sarcomeres, you'll find that in that position, you're not as strong as in the neutral position. And all that's showing is that muscle length is very intimately linked with the tension that it can develop. So that's everything that I wanted to cover in this tutorial. So just to quickly run over what we've been through, we've looked at the contraction cycle and seen how when the calcium arrives, it binds to the troponin and removes that tropomyosin off of the myosin binding sites that allows the myosin to flick over and form its cross bridges. We get the power stroke that pulls the actin towards the center of the sarcomere. And then we get ATP that binds and releases that myosin head. And then the ATPase enzyme then causes hydrolysis of the ATP molecule, forming ADP and an inorganic phosphate. And this process keeps happening so long as there's enough ATP around and you have enough calcium. And then finally, we looked at the length-tension relationship of muscles, and we saw how muscle tension is intimately linked with the length of the muscle. So thanks for watching this tutorial, and we'll see you in the next one where we talk about some of the aspects of metabolism in skeletal muscle.